Okay, let's go ahead and get started. In today's presentation, we're looking at the biological molecules of life. Particularly, these are the molecules that are covalently bonded that make up pretty much most of the body. In this section, we're going to look at the first one, which are the carbohydrates. When we look at the molecules of life, there are four basic categories. We have the carbohydrates, which are the sugars and starches. We have the lipids. You may not have heard the word lipid before, but this includes the neutral fats, which are the oils and the fats. In this case, we also have waxes and terpenes and phospholipids and something you've probably heard of before called a steroid. Those all belong to this group. Of course, proteins, which are built from amino acids. And finally, a group called the nucleic acids, which include things like DNA, and RNA, and ATP. All these are major biological molecules, and they're all classified as what we say is an organic molecule. Now, what the heck is an organic molecule? Organic molecules are molecules that always contain two elements. Those elements are always going to be carbon and hydrogen. Right? Even though carbon dioxide is a common waste product of the body metabolism and found in our atmosphere, it is not considered to be an organic molecule. It is considered to be an inorganic molecule because even though it has carbon, it lacks hydrogen. So organic compounds always have carbons and hydrogens in them. Right? That's what makes them organic. Now, when we look at the periodic table, we find that 92 of those elements are naturally occurring on the Earth and in the universe. All right? The additional ones, that you might see that we have 118 elements on the periodic table. A lot of those really heavy elements that are above 92 are actually produced in labs. They are such unstable elements that they usually only last a fraction of a second. They probably did exist a few times, but are so unstable that they break down. But if we look at the ones that we can find on Earth today, 92 naturally occurring ones. Now, living organisms are built from these different elements. But the remarkable thing is out of those 92 naturally occurring elements, life only uses 26 of them. Right? And the remarkable thing out of those 26, 20 of those elements are found in tiny, tiny amounts. They're found in trace amounts. We sometimes call them trace elements or trace minerals. These are things like selenium and copper and iron, right? And manganese and magnesium and zinc and selenium. Right? When we talk about trace, those are things that are found in less than 1%. So, most of our body is composed of just 6 elements. In fact, if we went and measured the mass of your body, we would find that 98% of what your body is made of is just 6 elements. Carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. We often use the abbreviation CHNOPS, C-H-N-O-P-S, as a representation of the 6 elements that really make up most of what you are. Now, this goes for all living organisms. If we look from the lowliest bacterium to the tallest sequoia tree, all right, we're going to find that those organisms are built from these same six, that most of the mass of an organism comes from those six elements. Now, the big remarkable thing is that if we go and remove the two least abundant ones in life out of those six, if we get rid of phosphorus and sulfur, all right, those four, carbon, hydrogen, nitrogen, oxygen, make up about 95% of the mass of an organism. So 95% of what you are, 95% of what a bacterium is, all right, is made up of just four elements: carbon, hydrogen, nitrogen, and oxygen. And those four elements are the principal components that we find in biological molecules, in carbohydrates, in lipids, in proteins, and nucleic acids. Okay, so the whole diversity of what you are, all the different chemicals and compounds and things, really comes down to a majority of them just based on four elements: CHNO, carbon, hydrogen, nitrogen, and oxygen. Okay, a little bit of terminology when we talk about organic compounds. Now, we all know what a molecule is. A molecule is when you have two or more atoms bonded together. If those atoms come from different elements, then we call that a compound. So not all molecules, well, all compounds are molecules, but not all molecules are compounds. For example, if we look at water, that's a compound. If we look at molecular oxygen, not a compound, but they're both molecules. All right. So we know what a molecule is. In organic chemistry, we find that a lot of these molecules are in big, long chains. All right. Oftentimes a whole bunch of repeating units, one after another after another. All right. So when we talk about them, they're usually a molecule that goes and bonds to another molecule, that bonds to another molecule, and produces this long, long chain. Think about a pearl necklace. All right, you have all these individual pearls, they're all strung together to make a pearl necklace. Each individual pearl, we can think of it as a molecule. What would we call that big string of molecules? Well, that big string of molecules in organic chemistry is what we call a macromolecule. Right? A macromolecule. In fact, here's a little example down here. This is a protein. Proteins are long chains of amino acids. So each amino acid that you see there is what we would call a molecule. When molecules are joined together, they form a larger molecule called a macromolecule. And macro, you know, just means large. So we have a molecule and then a larger molecule that's composed of those individual molecules. Now, we have another name for that. Right? Oftentimes when we look at a macromolecule, the fundamental unit, the fundamental molecule that makes up that macromolecule is often referred to as a monomer. So when we think about our pearl necklace, each individual pearl would also be described as a monomer. Monomer comes from a Greek word. Mono means one, and mer means part or section. So it literally means one part or one section. Monomer. Right? Now, if we have a chain of monomers, if we have individual molecules that are joined together to make a macromolecule, then we say that there are many parts or many sections, and that's what we call a polymer. You may have heard the word polymer before. Sometimes, I remember as a kid listening to television, and lots of times of advertisements would say, space-age polymers. So a polymer is just a long macromolecule. In fact, in this little picture here, what you're seeing is a type of carbohydrate that is found inside of our muscles, our skeletal muscles, as well as found in our liver. And this is a form of animal starch called glycogen. And if you look at glycogen, you'll notice that it's just one repeating molecule after another. That's the same molecule. That's glucose. That's just blood sugar. In fact, when we have excess blood sugar, our liver goes and takes that excess blood sugar and puts it into this large polymer that we call glycogen. And that's sort of our 24-hour food storage. We have about 24 hours of energy stored in our liver in this form as glycogen. And we find it in our muscles as well. So, some new terms: monomer, polymer, molecule, macromolecule. Now, in organic chemistry, it turns out it's very, very simple to understand how these monomers join together to make a polymer. Right? Whether we're talking about a carbohydrate, a lipid, a protein, or a nucleic acid, they are all built exactly the same way. They all have monomers. Those monomers join together to make polymers. Right? And it turns out they all use exactly the same chemical process for forming polymers. And I'm going to show you that next. That process requires the removal of water. So we call it the dehydration reaction. Right? Now, oftentimes when you look at these organic monomers, they will have attached to it, all right, a functional group that we saw before in water called a hydroxyl group. So if you look at this little graphic here, and you'll notice that on the monomer you have a hydrogen on one side, and then you have an oxygen and a hydrogen. That's called a hydroxyl group. So you have a little hydroxyl group there. Right? So if we're going to go, and let's say that it's your liver, and your liver is going to go and build glycogen from glucose, all right, what it does is really cool. As you can see, on one side of the monomer we have a hydroxyl group. On the one on the chain we have an exposed hydrogen. They come together and they form a water molecule. And the water molecule leaves. This is why we call this dehydration. All right? Dehydrate means to remove water. In fact, de means to remove. So when you hear about decapitate, all right, that means we remove someone's head. All right? So in this case, as that water is removed, then the monomer and polymer then form a strong covalent bond that joins them together. When we look at these polymers, every monomer is connected to the other monomer by means of strong covalent bonds. Right? So this is how all polymers are made. This is how all proteins are made. This is how all carbohydrates are made. This is how lipids are made. This is how nucleic acids are made. All right? So these monomers come together to form a polymer by using the removal of water, what we call dehydration reaction. Now, let's say that you decide to skip lunch today, and your liver will respond to that by going and taking its glycogen store and breaking off blood glucose. So break off those little glucose molecules from the polymer to keep your blood sugar up, all right, as your cells are using the blood sugar. Right? So how do we go and remove a monomer from a polymer? Well, turns out we have the opposite of dehydration reaction. We have to replace the water. And that's where we get the reaction that we call hydrolysis. Hydrolysis really comes from a word, hydro meaning water, lysis means to break or to burst. So literally, to break with water. And what happens is, in order to replace the hydroxyl group on the monomer that was lost when it became a polymer, all right, we have to use water. So in this case, you can see that water is added, and it goes to replace those little side chains, the hydrogens and the hydroxyl groups, to the monomers to be able to go and separate them. So in order to build a polymer, we use dehydration reaction. In order to break down a polymer, we have to use hydrolysis, where water is replaced. So water is removed to form a polymer. Water is returned when we break a polymer. Okay. Now, this is a very important table, right? This is a table that you need to treat like your significant other. Right? If you love somebody, you want to know everything about them. Right? You want to know everything about them. Right? I want you to treat this table like your significant other. And so what I want you to do is I want you to memorize, right? So you can just spit them out, the specific names for the monomers and polymers of the different categories of organic compounds. Now, there's a reason for this. To become someone fluent in biology, it's very important that you're able to recognize and recall the names for the different monomers and polymers of organic compounds, because we use them all the time. Right? Not only do we use them in this class, but from now on, you'll hear them and see them all over the place. So what I'd like you to do is I want to have it so that you know this inside and out, that this will always be in your back pocket. Right? I want you to know this as common as the joke that, what did I learn in biology? Mitochondria is the powerhouse of the cell. Right? I hope that's as strong as this one. All right. What's the polymer of amino acids? Polypeptide. Okay? So, what we're going to do is you're going to see this on several quizzes throughout the semester. I'll have this table, and I'll give you the class, all right, and you tell me what the monomer is and what the polymer is. Okay? And in our first quiz that has this on it, this is 8 points. Each blank there is a point. And to make it even more important, I count spelling. So you have to spell it correctly. You have to spell them perfectly. All right? So let's talk a little bit about them. The first one, carbohydrates. The monomer of carbohydrates are called monosaccharides. Monosaccharides. Mono, you saw, was one. If you guess, saccharide just means sugar, so it means one sugar. Turns out there are a couple types of polymers for carbohydrates. So I'm having you learn two. Polymer means more than one, all right? So in this case, we're going to learn the disaccharides. This is stuff like table sugar and milk sugar, all right? Disaccharides, 2 sugars, and then polysaccharides. Those are things like the starches. Glycogen, for example, is a polysaccharide. So many sugars, 2 sugars. Now, lipids is a very, very diverse group, but the ones that we're going to focus mostly on are ones that make up our long-term source of energy, the fats and the oils. These are called the neutral fats. The neutral fats have actually 2 monomers to them. So when we look at, animal fat or plant oil, we're going to find that those fats are composed of glycerol, which is 1 monomer, and fatty acids. So glycerol and fatty acids are the monomers that go and make the polymer, which we call triglycerides. Triglycerides are often what you measure when you get your blood tested. When you get your blood tested for cholesterol, the other thing that they'll often go and look for too is your triglyceride level. Now you know what that means. It's the polymer of fats and oils. Proteins. Proteins, the monomer is really easy. Those are called amino acids. You've probably heard about those. Right? Amino acids join together into long chains called polypeptides. Polypeptides. So amino acids and polypeptides. And probably the easiest one is going to be the nucleic acids. Nucleic acids, if you remember, DNA, RNA, ATP. Right? The monomer is just called a nucleotide. Polymer, polynucleotide. So nucleotide, polynucleotide. So we're going to go through each one of these in the next couple weeks, all right, and talk about them, but it's really important that you keep this in your back pocket, that you can pull this out at any time in your brain, because I see these terms every day, and for most people, I have no idea what they're talking about or what they're referring to. This is important for biology because you're going to see this over and over and over again. Alrighty. So we're going to start with our first ones. We're going to look at the carbohydrates. The carbohydrates are often referred to as the sugars and the starches. Carbohydrates always contain three elements. They always have carbon, hydrogen, and oxygen. Some might have a few other ones, but this is the general idea of carbohydrates. They always have carbon, hydrogen, and oxygen. And you'll find that the carbon, hydrogen, oxygen are always in the same ratio. Right? For every carbon, you'll have 2 hydrogens. So we have a 1 to 2 to 1 ratio. All right? So let's say we have 1 carbon, 2 hydrogens, 1 oxygen, a 1 to 2 to 1. Right? For example, if we look at glucose, blood sugar, its formula is C6. It has 6 carbons, it has 12 hydrogens, and it has 6 oxygens, a 1 to 2 to 1 ratio. So always that way. Now we've learned about carbohydrates for a long time. In fact, carbohydrates were first described way back in the 1700s. And the reason that we call them carbohydrates, if you go and you look at that formula there, that's just our simple little empirical formula, you'll notice that if you cover the carbon, what you got is a water. It's almost like you have a water molecule attached to a carbon. And this is where we get the name, all right? Hydrates of carbon. Hydrate refers to water, right? Hydrates of carbon. Now, where they got this idea comes from a really simple experiment. It's a little dangerous for us to do it because we use concentrated sulfuric acid. But if you take sugar and you put it in a test tube and you add strong sulfuric acid to it, what will happen is a vigorous reaction will take place. And what you'll get is a big tower of carbon. The sugar will break down into its 2 components. So you'll get this black carbon forming inside the tube, and you'll get this gas being released. And that gas is water. It's water vapor. It's steam coming off. And those are the 2 products that are found in sugar. That's all sugar is made of, is carbon and water. And hence the name hydrates of carbon. Now, the building blocks of carbohydrates we'll find as monosaccharides, right? And we'll talk a little bit about the polysaccharides as well. But turns out that carbohydrates are really the majority of our diet. Right? We are omnivores, all right? In a balanced diet, we eat carbohydrates, lipids, and proteins, particularly the neutral fats and the lipids. In fact, in our diet, about 60% of our diet in a balanced diet is going to be carbohydrates. Every culture has their food base or their staple as a carbohydrate. In Europe and America, our big thing is bread. Right? That's our grain. We like to eat wheat. We like to eat corn. Right? If you go to other places on the planet, you'll find that their big staple is rice. Right? Usually comes from a grain. Now, in other places, for example, if you went to Hawaii, the big carbohydrate staple has come from the taro root. Right? Or if you go to other places, if you go to the Andes Mountain, their big carbohydrate staple, potatoes. Right? So carbohydrates are 60% of our diet. And they're fantastic because they provide quick and easily accessible energy. Now, if we think about what organisms make carbs, the principal organisms that make carbohydrates are going to be photosynthesizers. So these are bacteria, what we call cyanobacteria, that are photosynthesizing bacteria. We have things called algae. Algae are photosynthesizing what we used to call protists, but they're single-celled eukaryotic cells. Yeah. And plants, all right? Those are the big three that are photosynthesizers. And if you think about it, they take this remarkable thing, a chemical process where they take carbon dioxide, some water, and a little light energy, and they convert it into sugar. That's where carbohydrates come from. They come from photosynthesis. Now, oftentimes, if you look at a plant has carbs, you bet, but it also has proteins and lipids and nucleic acids. Where do all those come from? Well, the plant has this remarkable ability to use that sugar, to use that carbohydrate as a raw material for building a fat or an oil or a protein or a nucleic acid like DNA or RNA. So oftentimes it is a raw material for building other macromolecules. Right? So we get those other macromolecules. We get those proteins. We get those fats and oils. Right? And we get those carbs from actually going and eating them directly from plants or from animals that eat those plants. The other thing, too, for these photosynthesizers, particularly when we look at the algae and the plants, right, and even the bacteria, we're going to find that carb, in fact, the special types of carbs, act as their cell wall material. And their cell walls are really important because that's what shapes their bodies. Right? So they're a structural material. Think about them as absolutely necessary for the architecture of those organisms. We live in a dry land, which is very weird, and it's a recent thing. You know, life's been on this planet for 3.5 billion years. Roughly only about the last 400 million years that life has been on land. Because land is rough to live on. You have a problem of gravity. Because in the water, everything's buoyant. And the other problem, too, is drying out. In water, you're surrounded by water. All right? On land, you have some problems with desiccation and drying out. So, especially for land plants, a great way to fight against gravity is to build a cell wall that turns your cell into a brick. A brick that you can stack and make an upright plant. So it turns out that these carbohydrates are fantastic as a structural material. All right. Now, the monomer for all carbohydrates are what are called monosaccharides. All right? And there are lots and lots of different types of monosaccharides. So let me tell you how they're classified. Monosaccharides all range from molecules that have 3 carbons all the way up to molecules that have 7 carbons. So there's a whole bunch of different monosaccharides, and they're classified into categories by the number of carbons that are found in their monomers. And you might notice when you look at carbohydrate names, there's a common suffix. That suffix is OSE, like ose. Think about glucose and sucrose and galactose, lactose, maltose, cellulose, amylose, ribose, deoxyribose. You'll notice that ose, that's a chemist's shorthand for saying, hey, you belong to a carbohydrate. Right? So, the number of different carbons classifies you as a specific type of carbohydrate. So, for example, let's say we look at glucose, fructose, and galactose, 3 important ones that we have in our diet, used for energy. Those all have 6 carbons. So they are classified as hexose sugars. Hex means 6. All right? So it's a sugar with 6 carbons. In DNA, we have a beautiful 5-carbon sugar that is called deoxyribose. And in RNA and ATP, we have another sugar that's called ribose. Right? Those 2 monosaccharides have 5 carbons. So they belong to a group that we call the pentose sugars. Pentose means 5. So 5 carbons. All right, the sugar with 5 carbons. So if we look at ones with 3, like you can see here, glyceraldehyde. Glyceraldehyde is a metabolite that we have as a carbohydrate that eventually loses a carbon and becomes ethyl alcohol. We'll talk a little bit about fermentation later in the semester. And the production of ethyl alcohol, the one step right before it is a 3-carbon molecule, a 3-carbon monosaccharide called glyceraldehyde. Right? So those are called triose, right? Because they have 3 carbons to them. So they are classified according to the number of carbons. Now, what we're going to be focusing on today is pretty much just the hexose. These are ones used for energy. Later on, when we talk about the nucleic acids, the DNA and the RNA, we'll talk about the pentose sugars. And you might notice there's one strange one called ribulose there. Ribulose, very important, one of the most common sugars on the planet you've probably never heard of before. Absolutely necessary in photosynthesis. We'll talk about ribulose when we get to the dark reaction in photosynthesis. So let's start looking at some of these hexose sugars, these monosaccharides. And we're going to begin with the principal food source for all of life. Living organisms live off glucose. All right? Glucose is what plants make. All right? Life really runs on sugar. And the simplest sugar, the one that is the food source for most organisms on the planet, is going to be glucose. Right? Now, glucose is also known as blood sugar, and it's also called dextrose. So if you see the word dextrose, you think, ah, okay, I know that. That's glucose. And if you look at the formula, the molecular formula, you'll notice that it's the characteristic C6H12O6. All right? That one should be easy to remember. All right? 612-6. All right? Now, you'll notice down at the bottom in the graphic that we have several ways to represent glucose, the structure of glucose. On your left is what we call a linear formula. All right? This is great because it shows you every carbon and it shows you what's coming off every carbon. Now, that form doesn't really exist in real life. Right? If we went out in nature and we took a sample of blood sugar and we looked at it with a really powerful microscope, we're able to actually go and look at organic compounds now. They wouldn't look in a linear stage. They would form a ring. Right? These monosaccharides will often go and, most of the time, go and form a ring shape. And you can see our little ring form there. Right? Now, the most important part, if we're an organic chemist and we're talking to another organic chemist, the most important part, the landmark, when it comes to a monosaccharide, is the carbons. Right? The carbons are the landmarks. If we're, for example, if we're talking to somebody about anatomy, and we talk about the eyeball, and we say, okay, the retina's in the back, the lens is in the front, all right? If we have a common language and we have a map of the eye in common, we can talk about it. Right? In the same way, chemists can talk about molecules by certain types of landmarks, if they have a common map in their head. And it turns out that with sugars, we're looking at the carbons. The carbons are the landmarks. So we number them. So you'll notice that on the right, we have carbon number 1. And then below that, carbon number 2, carbon number 3, carbon number 4, carbon number 5. And then if you look, coming off, almost like a little antenna, is carbon number 6. You'll notice that there's one corner in our ring that's not a carbon, and that's oxygen. Now, because carbons often form the corners of our ring, and they're so common, sometimes we leave the C out, and we just leave the corner. So in the next one, you see 1, 2, 3, 4, 5, 6. All right? That is a sort of a shorthand method to show that there's actually a carbon. There's a carbon right there. All right? Now, you'll notice the oxygen stayed. That's because it's weird. All right? It's not usual. The usual corner is going to be carbon. All right? So we often use that abbreviated ring structure. Turns out there are two forms of glucose. One called the alpha glucose and one called the beta glucose. Now, if you look at it, you'll notice, they look exactly the same, except coming off carbon number 1. So if you look at carbon number 1, you'll notice in the alpha glucose, we have a hydrogen on top and a hydroxyl group below. So hydrogen on top, hydroxyl group below on carbon number 1. If you look at beta glucose and you go to carbon number 1, you'll go, oh, they flipped. On carbon number 1, you'll see the hydroxyl group on top and the hydrogen down below. That's the only difference. That is the only difference between those two forms of glucose. But it makes a huge, huge difference when it comes to living organisms. Right? Turns out all the carbohydrates that contain alpha glucose, you and I can digest. All right? We can use it for fuel. All right? So these are all the plant starches that we eat. This is the animal starch that's stored in our liver. This is table sugar. This is milk, all kinds of sugars. All right, milk sugar. All right. This are all the dietary carbohydrates that we can all use for fuel. All use alpha glucose. Beta glucose, on the other hand, is indigestible to us. Think about trying to eat a cardboard box. You can't do it. You know, it goes in, but it's just going to come out as... Cardboard is built from a plant a plant carbohydrate called cellulose. And cellulose is nothing but beta glucose. And we cannot break the bonds there. We can't use it for energy. Beta glucose is a type of monosaccharide that is indigestible. Indigestible. So there's a big difference there. So all the carbohydrates that we can digest, that we can use for fuel, alpha glucose. Alpha glucose. And the big difference between them is what you see off carbon number 1. So hydrogen on top, hydroxyl group below on alpha, hydroxyl group on top, hydrogen below on beta. Here's our other one. This is fructose. Fructose is often referred to as fruit sugar. It also has another weird name. Sometimes you'll hear it referred to as levulose. All right? But basically what this is just fruit sugar. Now, you'll notice that it has exactly the same formula. C6H12O6. Exactly the same formula. But you'll notice that the structure is different. Right? You'll notice that on carbon number 2, there's a double bond with oxygen. Now, if we looked at glucose, we would find that it's carbon number 1 that has the double bond with oxygen. That little change is significant. Look at the ring structure. It looks like the pentagon. It has 5 corners instead of 6. This is very easy to recognize. Fructose is super easy to recognize because it has that pentagon shape to it. Now, turns out fructose, which is very common, obviously in fruits, if we did a taste test between fructose and glucose, we would find that fructose is much sweeter. It tastes sweeter. Wow. Since it tastes sweeter, food scientists discovered a long time ago that if you're going to sweeten a food and you use regular table sugar, which is sort of half glucose, half fructose, right, you have to use a certain amount in order to get the sweetness. But if you replace that table sugar with nothing but fructose, right, it can be sweeter, but with less sugar. Does that make sense? So you can get the same level of sweetness with just fructose than you can with a combination of fructose and sucrose and glucose. That means you don't have to use as much. And if you don't have to use as much, it's cheaper. So food scientists for a long time, and they still do this, actually go and make high fructose corn syrup. It's derived from corn. Basically what they've done is have this concoction that is nothing but fructose. which is wonderful. Now that it's nothing but fructose, wow, it's going to be sweeter, right? And so you don't have to use as much, so you don't have to spend as much. So a lot of our processed foods are sweetened with high fructose corn syrup. Right? For years, when I would go to Winn-Dixie and go down the soft drink aisle, you could not find a Coke or a Pepsi or Dr. Pepper, right, that didn't have high fructose corn syrup. Now, there's a little swing back. People are nostalgic for the time when it had real sugar in it, when it had real sucrose in it. Right? And so nowadays you can find versions of it, Pepsis and Cokes, and here's a Mountain Dew and Dr. Peppers that have real sugar. Right? So now our last monosaccharide is an interesting one. This monosaccharide is found in milk, but it's not by itself in milk, but it's part of the sugar that's found in milk, and it's called galactose. You might be wondering, well, galactose sounds a lot like galaxy. You'd be right. It comes from the same word root. Think about what galaxy our solar system is part of. The Milky Way. Right? Galaxy refers to our galaxy, the Milky Way. Galacto means milk. Right? So this is one of the monosaccharides that is part of lactose, which is milk sugar. This is rarely found free. All right? In fact, very quickly, when we go and ingest galactose, we convert it to glucose. All right? So this one is rarely free, it's just plain galactose. But you'll notice that it has the same formula as fructose and glucose, C6H12O6. The other interesting thing about it, too, is that it is structurally very similar to glucose. And I bet you can pick it out. If you look at carbon number 1 and carbon number 4. Right? So let's look at alpha glucose. You'll notice that at carbon number 1, we have a hydrogen on top. Carbon number 4, we have a hydrogen on top. Right? That's alpha glucose. If you look at galactose, it's just the opposite. You'll notice that we have a hydroxyl group on top at carbon number 1 and a hydroxyl group at carbon number 4. That's the difference, is that 1 and 4 have flipped. Now, I have a risque way of remembering this. And once you've learned it, you'll probably never forget it. Oftentimes, if you go and you listen to medical students, how they go and memorize things is they often will try to make it nasty. It's easier to remember nasty things than it is fairly clean things. So I apologize. I hope I don't offend anybody. But if you look on that, you can say, oh, galactose. That's the molecule where the H's are on top. The hydroxyl groups, HO. Right? So you can see that on carbon number 1 and carbon number 4, it's the hydroxyl groups that are on top. The H's are on top. And that's how I remember it. And hopefully, you'll never forget that one. Okay. Now, we have a name for molecules that have the same formula, but different structures. And they are isomers. You'll notice in my little diagram here that all three of these monosaccharides, glucose, fructose, and galactose, all have exactly the same molecular formula. All have 6 carbons, 12 hydrogens, and 6 oxygens. But they all have different properties. And those properties, for example, sweetness and usability, are all basic differences in their structures. So molecules that have the same molecular formula but different structures are called isomers. Isomer comes from words that mean iso means equal, and mer, as you know, means part. So equal parts, same molecular formula, but different structures. And that makes all the difference. So what you're seeing here among the monosaccharides, the hexose ones that we've talked about, all three of them are isomers of each other. Okay, now if we go and we join two monosaccharides together, we form our first polymer. And we're going to go and look at what are called disaccharides. Di means two. We often go and feast on these. All right? These are ones that are found in our food. In fact, we find out that for living organisms, particularly plants, all right, well, let's just say plants. Plants often transport their sugars as a disaccharide. This is how plants often go and transport their sugars as disaccharides. All right. So we're going to start off with a first one, and this one's really, really simple. All this one is an alpha glucose bonded to an alpha glucose. This one is called maltose. Maltose actually is a sugar that's found in germinating grains. Now, when we think about grains as a type of seed, for example, corn is a grain, wheat is a grain, oats are a grain, all right? Rye is a grain, barley is a grain. All right? We think about grains as a major source of carbohydrates around the planet. A lot of organisms, including humans, most of our diet comes from these grains. Now, if you think about what a seed is, like a grain seed, it's basically inside the seed we have a tiny little embryo, which is going to become the plant. That's the baby plant. And the lunch that the mama plant made for the baby. And that's going to have some oils in it. It's going to have a little bit of protein in it. But a lot of it is going to be starch. And starch is a polysaccharide. It's lots and lots and lots of alpha glucose in long chains. Now, maltose is basically just two alpha glucoses joined together. How do we go from a big polysaccharide where we just have long, super long chains of just alpha glucose to just a pair, a bonded pair of alpha glucose? That's maltose. Well, it comes from enzymes. Right? So what happens is when the conditions are right, a seed will start going and breaking down the starches that are found inside the seed. We often call that the endosperm. So it'll start releasing these enzymes and it will go and start breaking down these starches into maltose. And sometimes we do that on purpose. In this little picture here, you see a strange room, and what that is rye or barley, right? And you can see that it's all there. They're raking it and everything. Well, what they'll do, in order to go and generate maltose, is they'll take that seed, that grain, and they'll put it in a place that would seem perfect for a grain to germinate. So what happens is the grain starts going and releasing these enzymes, and it starts growing little roots and things like that, and it starts breaking down this starch into much more palatable maltose molecules. Now, once we got enough maltose in it, then they stop the germination. Right? And then they kill the little embryo, and then they feed the seed to yeast. Yeast love maltose. Right? And maltose is the food source for the yeast, and the yeast will produce alcohol, and this is how we get single malt whiskey. Right? Have you ever heard of malt whiskey? Right? It comes from the malt, which is basically you germinate a seed, the seed breaks down the starch into maltose. Right? So if you heard of malted milk, right? Malt balls like Whoppers, right? That's what that is. Rich in maltose. Now let's look at the molecule here. You'll notice to form maltose, we have a dehydration reaction. So we have hydroxyl group and a hydrogen joined together to make water. So dehydration. Right? And it forms a strong covalent bond that joins both alpha glucoses together. Now, you'll notice that we have a special name for the covalent bond that joins these two monosaccharides together. And we'll find that every group of organic compounds, the lipids, the proteins, the nucleic acids, and the carbs, all have a specific, unique name for the bonds, the covalent bonds that join monomers together to make a polymer. And for carbohydrates, that name is glycosidic. So it's called a glycosidic linkage. Sometimes you'll see it as a glycosidic bond. I like linkage. Glycosidic linkage. You'll also notice that when you look at it, there's numbers in front of that. The numbers represent the carbons that are joined together. So for maltose, you'll notice that it's referred to as a 1-to-4 glycosidic linkage. 1-to-4 glycosidic linkage. That means that it's bonded between carbon number 1 and carbon number 4. Now we add one more thing to this. What type of glucose is this? This is alpha glucose. So that chemical bond, its full name is alpha, because of the alpha glucose, alpha 1-to-4 glycosidic linkage. That's how we would describe this one. Alpha 1-to-4 glycosidic linkage. So that's maltose. The next one is something that we eat every day. And it's table sugar, it's cane sugar. In fact, this is one of the major transport carbohydrates in plants. If you think about maple, all right, maple sugar, think about sugar cane and sugar beets, all right? This is how that is transported. It's transported through a plant as sucrose. Right? Now, sucrose is a combination of two different monosaccharides. We have alpha glucose bonded to a fructose. It's very easy to recognize because fructose looks like a pentagon. Right? If you see how they bond together through dehydration reaction. All right, if we want to break that bond, hydrolysis. In this case, when we look at sucrose, table sugar, cane sugar, maple sugar, all right, all the same thing. All right. The type of bond that we have here, and it's a little weird. Right? It comes from a bond from the first carbon on alpha glucose, and it bonds to the second carbon, as you can see there. The numbering is a little bit weird on fructose, so they call that carbon number 2. Carbon number 2. So we would call this glycosidic linkage, this bond, alpha, because it's an alpha glucose, 1 to 2 glycosidic linkage. That's what we would describe it. Alpha 1 to 2 glycosidic linkage. Now the last one is a really fascinating one. And it has a lot of really cool biological manifestations. This is lactose. This is milk sugar. About 3.5% of whole milk is going to be milk sugar. Milk sugar is secreted by lactocytes, special cells that are found inside the breast, inside the mammary glands. And this milk sugar, which is produced within these lactocytes, is incredibly osmotic. What we mean by that is water is attracted to it. So if we think about milk, right? Milk is mostly water, about 88% water. So one actually goes and produces that water as these cells secrete it. These cells are actually using lactose as a way for water to be attracted to it so that we get this fluid. For infants and for baby mammals, all right, this is their biggest source not only of nutrition, but also water. This is how baby mammals drink. So that's why we have lactose there. It's fantastic as a way to attract water. Now, lactose, as we call it in milk sugar, is composed of 2 monosaccharides. You'll recognize that leading the way here in the front is galactose. In fact, the form that we learn with the hose on top is what we call beta-galactose. Beta-galactose. And it forms a covalent bond with glucose. Now, as you can see, it's a bond between carbon number 1 on beta-galactose with carbon number 4 on glucose. So it's a 1 to 4 glycosidic linkage. But you'll notice something a little odd. Since this is a beta-galactose, what's going on is we call this beta 1 to 4 glycosidic linkage. A beta 1 to 4 glycosidic linkage, because this form of galactose is a beta form. Now, you'll notice that the bond looks a little weird. It looks like an N. That is a standard way to show you that in real life, when these things bond, the second molecule, the second monosaccharide, in this case alpha glucose, is going to be upside down. It's going to be upside down. So rather than draw it upside down, which would be a little bit tricky, what they'll do is they'll actually go and make the bond look like an N. So that N says that in real life, that glucose is flipped upside down. So that's what you're seeing there. Now, lactose is important. Mammals are the only animals on the planet that feed their young milk. So every mammal grows up on milk. But a very, very strange thing happens only with humans. As infants, all mammals produce an enzyme. This enzyme is called lactase. Right? Enzymes are often named for what they work on. So lactase, anytime you see ASE at the end of a word, think to yourself, oh, I bet that's an enzyme. That's a common suffix that chemists use for enzymes. Right? So here we have lactase that goes and breaks down lactose into galactose and glucose. Now, turns out that once we get done weaning, all right, once we grow up, every mammal, except for humans, every mammal loses that production of lactase. So an adult mammal cannot go and break down lactose. In fact, if they do that, they will actually go and get cramps and diarrhea and lots of flatus, which is intestinal gas. Right? Because they have an insufficient amount of lactase to go and break down the lactose. Except for humans. Now, it's not all humans. About a third of people on the planet have what's called lactase persistence. And what that means is, in adulthood, they still are producing the lactase enzyme so they can drink milk and other dairy products and break down the lactose and not suffer from the diarrhea, the cramping, and the flatus. So, I'm going to show you, in fact, linked to our little lecture here, is a video all about the development, the evolution of lactase persistence in humans. And I hope you watch it because it's absolutely fascinating. There's some great biology in it. Okay. Let's go ahead and stop there. In part 2, I'll talk about what we call the polysaccharides, and then we'll be done with carbohydrates.